Hello students, I am Amit. Today let me welcome you for another session of Chapter at a Glance. Today we are discussing molecular basis of inheritance, one of the central and most important chapters. The importance of this chapter can be understood by the basis that how many questions are appearing every year in this particular chapter or from this particular chapter. Let's start with that. So when we see this question, so on an average we are seeing around 7 to 8 questions are appearing from this particular paper or from this particular chapter. 7 to 8 means that it is around 8 to 9% of the entire biology paper that you are going to see. Numbers have gone up and down. We have seen 6 questions also. I think 2023 was the time when we, 2024 was the time where we actually saw the less number of questions, but on an average we are seeing 8 to 9 questions almost every year. That's why I have lowered the average around 7 to 8 questions. Remember that 2 zoology topics, which are Human Genome Project and the DNA Fingerprinting, both of them are also part of this particular session. Easily every year 8 to 9 questions are always seen in this particular chapter. Let's see that what are the other averages that we are seeing and all year on year, what is the importance of the chapter. So in 2023 I had 8 questions, 2024 6 questions, 2025 8 questions. Remember that in the mix if I bring in other questions, because why I am saying other questions, because there are some sections which are appearing from other chapters. Let's say questions on polyribosomes or polysomes, which is a question part of cell. Question on chromatin, which is also mentioned in cell chapter. Biomolecules, where nucleosides and nucleotides are mentioned. Their diagrams are actually mentioned in that particular chapter. So if I bring those into the mix, so these are called overlapping sections. Those will take its number easily to 8 to 9. Around 10% of the entire NEET biology section is going to appear from this particular chapter. You are not escaping this chapter. You have to do this chapter if you want to score, if you want to become a doctor, if you want MBBS seat. This is your way through. That's why we are going to give this chapter that importance. First we'll discuss the schematic or what is there in the chapter. Then we have made some tools for you. We'll use those tools so that you can have a quick revision of this chapter. Remember that. Understand the focus of this video. We are not discussing the chapter in depth. We are discussing what has appeared before in NEET exams, what is important. At the same time, we are going through a quick revision. So we are not discussing the small minute details that might occur in the chapter. Important chapter, you don't have to miss any detail. But this is something that, okay, my NEET exam is just round the corner. Let me do something about it. Let me have a quick revision of the chapter. Because this happens that we miss certain points here and there, let's try to brush up on those points and let's see what happens. So we made a small image or a small mind map for you. It does have some sections. If you see this, you will have some sections available in which we have mentioned DNA Fingerprinting and Human Genome Project also. Remember that they are part of zoology section, but they are mentioned here because they are part of the chapter in NCERT. So when we are starting with the chapter, we are starting the chapter with the genetic material itself, with the DNA itself. NCERT, before even telling that how DNA became a genetic material, it talks about the structure of DNA. How DNA look like, what is the bonding look like, what is a glycosidic bond, what is a phosphoester bond, diester bonds, all those parts are given at the starting portion. So when we are discussing, so DNA, what are histones, what is double helical model. Yes, so in this there are 3 scientists which have to be mentioned. One is Wilkinson Franklin, one is Irwin Chargaff, and one is going to be Francis and Crick. These 3 groups are there. Important groups are there, but they are discussed here only. Questions on them when they appear later, we'll discuss them along with that. And what are the differences in their packaging, in which we discuss about eukaryotic and prokaryotic structures, where nucleoid is mentioned, as well as in eukaryotes you will have the chromatin mentioned and how the chromosomes are being formed, that is mentioned. Next part of the chapter will take you to search for genetic material. In the search for genetic material, the NCERT part starts with that what are my expectations with genetic material, what it should be, what it should not be, they discuss a bit about what is DNA, what is RNA, what is the difference between DNA and RNA, and at the same time they discuss how DNA came to be a genetic material. In this, you have key 3 experiments which come, in which you have transforming principle by Griffith, you have biochemical characterization of transforming principle by McLeod and McCarthy, and then unequivocal proof that was given by Hershey and Chase. This is a mandatory question. I am getting one question from either of these 3 experiments. Either it is in the form of the matching, either it is in the steps formation. You will find the question on this particular portion. So when we have discussed all the experiments and all, you come to the conclusion, okay, DNA is a genetic material, or to be very frank, DNA is a better genetic material than RNA. That is the conclusion that we draw from this particular event. Now when we discuss the next part, just before the start of the processes or the central dogma, we have RNA world, in which we have to discuss that how RNA was the first genetic material, how the processes of translation and genetic material or like DNA has evolved around RNA. So that is an important aspect. RNA world have made questions before. When we discuss that, what kind of questions have appeared from them, we will discuss along with NCERT. Now the first thing of the central dogma, we have what? We have replication. In this, the first part is semi-conservative nature of replication. Mearson's-Stahl experiment, if I see from last few years, I am seeing questions on a regular basis. Let's see that if I say 4 exams, 3 exams question Mearson's-Stahl experiment will appear. It's such an important experiment. It's such an important question. It makes numericals also. It makes conceptual parts also. In replication, we have to discuss what is origin of replication. Remember that this is the same origin part. There's a small line again, we'll discuss that line in NCERT also, which connects it to the biotechnology section. What is a replication fork? What are the DNA polymerases used? What is the leading and lagging strands? Very, very important aspect. What are the directions of those? And then what is the activity of ligase? I'm sticking to NCERT. There are other things, helicase, Okazaki fragments, other terminologies and all which might appear here and there. Primers, all those things and all, they will appear. But remember that we are sticking to NCERT. When you are brushing up towards the end, brush up with NCERT, brush up with, stay with NCERT, so that any question, if I see the last 5 exams, I have seen very NCERT-centered topics. Yes, we have seen topics which have appeared away from NCERT also, but none of the questions were in such a way in which you can't do the questions only with NCERT. I have seen, let's say I'll give you an example, a question on Tata box appeared, or a question on primers appeared. They are not mentioned in NCERT as such, but they were part of the matching. If you do other matchings correct, then this matching is already done. NEET is maintaining itself around that when you have NEET exams, NCERT is the core of that exam. After we have discussed replication, then we directly jump into the transcription section. In the transcription part, very, very important part is going to be transcription. One of the most asked question is a transcription unit. What it is made up of, what it look like, what is the coding strand, leading strand, coding strand, and what is the template strand, all those parts. So promoter, genes, template and coding strand, these parts. In this, we have to discuss the types of genes also, monostonic genes, polystonic genes, all those terms also appear. And this split gene arrangement also appear. Then you have prokaryotes, which have only one RNA polymerase, eukaryotes, which have RNA polymerase 1, 2, 3. Again, very, very important question, this part. I think in last 10 years, the most repeated question appear from this: what does RNA polymerase 1 do, what does 2 do, and what does 3 do? There is no skip in this question also. You will see year on year the question is appearing from that particular section. When we have discussed the transcription, we will jump to the next part, that is the genetic code. Important section, genetic code. If I remember one exam, I think 2-3 years back, there was one exam which had around 3 to 4 questions only from genetic code section. It is such an important part. 100% you are going to find a question on genetic code section. Now either the question can be on scientists, either it can be that what is the start codon, stop codon, all those things. But when we are discussing who deciphered the genetic code, so George Gamow, Nirenberg, Hargobin, Khurana, all those. In the salient features, what is the triplet nature, nearly universal nature, degenerate nature, ambiguous, non-ambiguous nature, all these have to be discussed. Then you have mutations. At the end of genetic code, a small portion of mutation section is given. Remember that this mutation section explains the parts in zoology and principles of inheritance, in which you have done certain things like sickle cell anemia and all those. But the explanation of them exists actually at this point. So in mutations, frame shift and point mutations, important part. Frame shift have made good numericals over the time. We'll discuss those also. Then you have tRNA's adapter molecule, inverted L model, important one, Cayman-Klug model. Before that, tRNA had cloverleaf model that was given in NCERT. Remember that one is 2-dimensional model, one is 3-dimensional model. Both of them are correct models, but tRNA is also explained in NCERT. But after the books in 2018, we have change in the structure given in NCERT. Apart from this, now we have to discuss the next part, that is going to be translation. Very, very important section. I'm saying important so many times, but in reality, this chapter is like that. If you are getting 8-9 questions from one chapter, I will expect question from every section. There's nothing to leave. You have to do everything in this particular section. So when we are discussing translation, in translation part, we are discussing activation, initiation, elongation, how it is done. Very, very important lines in NCERT. What is the first phase of translation and what is the first step of translation? Two important things, two differences which are there. We'll discuss those. Very, very important section, very interesting section also. Then you have initiation path, elongation and termination is also there, and then you have the catalyst section, in which what is the activity of ribosome, ribozyme, all those sections are very, very important ones. Towards the end, remember that towards the end of the botany section, important section of Lac Operon or important section of regulation is mentioned, in which what are the stages of regulation, that is an important aspect. And in NCERT, only one kind of regulation is explained, that is non-constitutive version that is found in operon systems. So in that, one inducible operon is mentioned, that is Lac Operon. In this you have Jacob and Monod gave it, what is a repressor, inducer, genes, how it works. Again, 100% question, very, very important question. Lac Operon makes a question every year. Very important part. Lac Operon is going to give you one question, not very typical question. Good questions come, NCERT lines come, but remember that a small part of the Lac Operon mixed with the biotechnology section, where you talk about LAC-Z gene, in which you do blue-white colony selection. The roots of that are actually seen in the Lac Operon section only. So this is an overview of the chapter, just to tell, okay, these are the things, so that nothing is missing out. After this, what we are going to do, so what we have done for you, see, it's a big chapter, it's hard to revise. We have made certain tools for you, we had made certain tables for you. These tables, these tools will help you to quick revise anything. See, in the entire 8-9 questions that I am discussing from this particular chapter, my expectation is that around 3 to 4 questions are going to be matching type or direct factual questions. So all the facts, like scientist name, their experiments, how did their experiments, and the years in which they are appearing and all, what is the key factor in that experiment, all those we have put into factual tables. So we have made actually 2-3 tables or 4 tables, so that just before 5 days are remaining, I have to revise this chapter, very, very important chapter. How to do that? I am going to do that through these tables. If our information is absent in NCERT, we have also not copied that information here. If you see the years, so these are all the scientists which are given in the NCERT section. Here all the years are also mentioned. Deliberately left those sections empty where the years are not mentioned. If you find those years, you want to write the years, you can find them. This is an important section. If you want to pause the video here, you want to look at it, zoom out the video, look at the facts and all that. Easy facts are mentioned here. We have mentioned the smallest things, so that whatever question comes, I can answer those if it's a factual question, because match the following is a very common question. Let me start with first. First is Frederick Mesher. Frederick Mesher identified the DNA as an acidic substance, so the word nuclein as established by him. Then you have Mr. Frederick Griffith, who actually gave transformation of bacteria. Next one is Avery, McLeod and McCarthy. They worked on the transforming principle part, in which they actually gave biochemical, or they explained biochemical characterization of the Griffith experiment. So they actually proved that the transforming principle is actually DNA. Then you have Alfred Hershey and Martha Chase, which gave unequivocal proof. This is the most heard word in this particular chapter. Then you have Watson and Crick. They have done so many things, but we have mentioned them. They gave what? They gave double helical model. They've also suggested the scheme for replication. Though Francis Crick comes at different places also, like he comes around that central dogma. This was Francis Crick, and then who suggested the adapter molecule also. In that you also have Watson and Crick mentioned or named there. Then you have Morris Wilkins and Rosalind Franklin. They are known for X-ray diffraction data, which is used by Watson and Crick to give the helical model. Irwin Chargaff, very, very important person. Mr. Irwin Chargaff, he gave base ratio constant that adenine is equal to thymine and guanine is equal to cytosine, and pyrimidines and purines are equimolar in nature in a double-stranded DNA. A very important aspect. Then you have George Gamow. George Gamow, who gave, so many students think that he was the one who proposed the genetic code. He did not propose a genetic code. He proposed genetic code as a triplet. That is an important aspect. Hargobind Khurana, he is known for synthesizing the RNA molecule in a cell-free system. Then you have Marshall Nirenberg. He is known for protein synthesis. So for RNA synthesis, Hargobind Khurana. For protein synthesis, we have Marshall Nirenberg. Then you have Severo Ochoa. This question also came once. Polynucleotide, what is the name of Ochoa enzyme? That is polynucleotide phosphorylase, important enzyme. Matthew Michelson and Franklin Stahl, they are known for semi-conservative nature in prokaryotes. While when it comes to eukaryote, that is Taylor et al. And proved that DNA chromosome replicates. This is actually going to be in eukaryotes. If the word chromosome is used, go for Taylor et al. If the word bacteria, prokaryotes, so generally semi-conservative word is used, give preference to Mr. Michelson and Stahl. And then you have Francis Jacob and Jake Monod, which is known for elucidation of the Lac Operon. Very important section. Next one is Frederick Sanger and Alec Jeffrey. You have done them in zoology already. There are different sections. They are part of Human Genome Project and the DNA fingerprinting. Can we go to the next part? So this is the first tool. Let's go to the next tool. So I have already told you about the experiment. I have already told you about the scientists. Now the steps given by these scientists, that is also an important aspect. So when it comes to those steps, what we have done in this table, the key points we have already discussed, that what the contribution of that scientist is. In this we have taken and we have specifically named or specifically put all the things which have been done in that experiment also. A very quick revision is available. Let me just go through one or two things and all. I think one can be Griffith experiment. Straightforward things are given. First point, it is about that he grew streptococcus and he found one is the smooth type and one is the rough type. Second one, then he injected the mice. Then he did talk about, then he took the mice and he gave SS strain, and we saw that the mice died, and when the mice injected with the R strain or the heat killed S strain, it actually survived. Next one is injected a mixture of heat killed S and the live R. We actually saw the mice dying, and you have recovered S bacteria from the autopsy or from the dead mice. So all in all, R strain transformed into S strain. This is what this experiment tells. Let me take another experiment. Let me take Wilson-Stahl experiment for that matter. In this, he grew E. coli for many generations in a medium containing heavy nitrogen. That is the first step. Second one, transferred the cell into the medium with normal nitrogen, and for many generations you are going to grow it like this. And then for every generation you have used cesium chloride gradient so that you can separate out the light and the hybrid and the heavy DNA. That is the next step. And then you have observed the DNA extracted, that after in 20 minutes you have hybrid DNA, and 40 minutes you have light and hybrid DNA, and so on. These are the parts. I have discussed only two. Just go through them. If you want, you can pause it right now and you can look into these steps. Only NCERT parts are important. Sometimes you have chronology questions which are appearing from this, in which they will give you 5-10 steps and they will ask you to put them. Very important question. Let me just do that Hershey-Chase experiment also. So the bacteriophage experiment. Now issue is that in bacteriophage experiments there are 3 steps which are given. One is the infection, and the second one is you start with infection, then you have slow centrifugation, then you have proper centrifugation, or you have a detachment step in the middle. But many students miss something actually, the growth part. If you start in the NCERT, how the virus are grown, that part is also mentioned. It is not in the diagram, but how we are going to get radioactive viruses, that is actually explained also. That is the first step. So important steps are here, very, very important parts are there. This is the next one, important one. Let's go into the next part. Next part is about the processes. Again, we are here not trying to go into the depth of the processes, we are trying to get the crux of the processes. All of you already understand the replication, transcription, translation, but here we are just try to get the essence of what was they truly in NCERT. So that's what we are trying to do. The next part is talking about replication, transcription and translation. This is the first slide. In next slide, what we do, we have again discussed these processes, but with the lens of prokaryote and eukaryote. That slide will be the next one. We'll discuss that slide also. Okay, so in every process, DNA replication, transcription and translation, we have a preparatory phase. Now, what is that preparatory phase? For case of DNA replication, we have the formation of DNTPs, that is deoxyribonucleoside triphosphates, and their dual purpose, here they are acting as substrate as well as the energy source. Similar thing is happening in transcription. They are also, the nucleoside triphosphates are there, or we can use the word ribonucleoside triphosphate. Deoxy word is not there. They are also using substrate. Remember that they are also what? They are also energy source. So this is nothing different, but they are also what? They are also energy source here. Next one, translation. We have an important portion here. So amino acids are activated. This activation is also named as what? Amino acylation of tRNA, where a tRNA is attached to its cognate amino acid. Important one, very, very important section. Now, in all the cases, let's talk about the initiation. When we discuss the initiation part, initiation needs origin of replication here, very important section. And this is the origin. Without origin, can the replication happen? No. So in eukaryotes, you have many origins, but in case of prokaryotes, you have only one ORI site. Now, a small opening happen, and then replication fork is made. Replication fork is an important part, which is made due to the opening of the DNA. Then you have RNA polymerase part. All the steps in this you have an enzyme called unwindase, which is helping me in this, or helicase. In this, all the things are done by whom? They are done by RNA polymerase. There was a question also in transcription, who does the opening of the DNA? Who is doing that? It is actually RNA polymerase who is doing it. All the steps are also there. So the key part that are given here, what is going to happen, that the initiation factor is also mentioned. Remember that initiation factor is only seen in prokaryote, or sigma factor of initiation is seen in prokaryotes only. Then you have translation in NCERT. So this is the first part in which the activation is happening. In this, the actual start is happening when you have mRNA, or the ribosome binds to the mRNA at the start of the codon AUG. This is recognized only by the initiator tRNA, and this is the initiation portion in case of the translation section. Next part is elongation. Here, what is the key enzyme? DNA dependent DNA polymerase is the key enzyme. In this, the polymerization is strictly in one direction, that is 5' to 3' direction, line from NCERT. Now, the continuous part is on which template? Is on 3' to 5' template, while the 5' 3' template will have discontinuous version. That is what is mentioned in this, that is the unique part. When it comes to RNA polymerase, only one strand undergoes into transcription, that is called the template strand, that is actually contributing, but coding strand doesn't participate. So DNA polymerase polymerizes in a template dependent fashion following the rule of complementarity. This one and this one, both of them follow the word, very important word, the word is complementarity. Somebody asks that what is the basis of replication and transcription? That is actually complementarity of bases, but a small change happen here in translation. Now, we are not going into the detail of the process, but the complex is composed of an amino acid linked to tRNA sequentially bind to the appropriate codon. So one by one, what is happening? One by one, the codons are adding up. Where tRNA add to the what? To the anticodon, and the ribosome move one by one, and it keeps on adding polypeptide chain. The catalyst here is ribosome itself, which is acting as a ribozyme, and the formation of the peptide bond happen. Here, the word was complementarity. Here, the word is collinearity. codon anticodon relationship is based on complementarity, but amino acid to codon relationship is based on collinearity. Important aspect, remember that, that's a different part. And towards the end here, there's nothing very specific that is happening towards the end, but at the ending you will have ligase working here. Ligase is going to join the discontinuous strands. NCERT have not given the concept of primers as such, but RNA primers are an important aspect of this particular part. Next one is going to be the specific ending of the transcription. Now one question here, students mix up transcription and translation ending. Remember that when it comes to ending of transcription, we have something called a row factor, because in both the case we can use the word termination, and termination creates confusion in your head. Termination of transcription or RNA synthesis is going to be done by row factor, where in this case you have something that is the release factor that is going to, here this is release factor, and here it is going to be the termination factor, that is the row factor. So that's, remember that both of them can be called sometime termination factors, but one is release factor and one is row factor. Important portions. This is an ATP dependent process also, this is also a ATP dependent process. Again, this is just the essence. Now I understand the starting, or let's say before starting, the middle and the ending of the processes. Questions generally appear from this particular, the confusions generally happen in those sections only. Let's go to the next section. We are going to the next part. I told you already that here we are going to discuss the similarity and dissimilarities with respect to prokaryote and eukaryote, because NCERT keep on telling these things in the middle and they are very hard to separate out. Though most of the processes, whenever they are explained, they are mostly explained around prokaryotes. Like eukaryotes are not given precedence in NCERT, but still let me just take out this part, very important table. It gives you idea in what is happening. So now in this you have replication, where you have the timing and the location. It is a definite region of DNA termed as origin of replication where it is happening in case of eukaryotes. So takes place in S phase of the cell cycle. S phase is not mentioned in bacteria. Bacteria don't have a S phase, and also chordate. They have something called MOR, multiple origin of replication, which are found in eukaryotes. What is the similarity? In both they follow semi-conservative mode. They both require DNA dependent DNA polymerase. Remember that in case of prokaryote you have 3 types of DNA polymerases. Here you have 5 types of DNA polymerases available. Next one, in transcription, what are the enzymes? Here you have only single DNA dependent RNA polymerase. This can be written as DDRP, or sometime books also write it as RNAP, RNA polymerase. This is only one type, where you have 3 RNA polymerases present here, which is 1, 2, 3 type. Remember that in a line in NCERT, I'll bring up you to that line also. There's a small difference given for RNA for polymerase. They say in a eukaryotic cell there are actually how many RNA polymerase? 4, because 3 are to this type, RNA polymerase 1, 2 and 3. At the same time in organelles in mitochondria you have prokaryotic version also. So in a eukaryotic cell how many RNA polymerase are there? 4 RNA polymerase are there. Important question. So both the processes are governed by the principle of complementarity, also already given. Both have them the adenosine and all that. The segment of DNA, only one of these strand is copied. Both the things are similar. Now what is the gene structure here? Here the transcription, the unit is mostly polycystonic, important portion. Though when it comes to this you have something called a split gene arrangement. They are monocystonic for the first, and second one is split gene arrangement, that is already there. Though both of them have promoter, structural gene and terminator, though there are other factors which might be there in eukaryote. Eukaryotic transcription according to NCERT is a complex process. Next one is RNA processing. The RNA does not require any processing to become active, but here you need processing, in which you have splicing, capping, tailing to become active RNA. So from hnRNA to mRNA you have to do that. Next ones are the translation, coupling and location. So how they are connected to each other? So if you see this, because there is no separation of cytosol and the nucleus, the transcription and translation take place. So we use the word what? We use the word simultaneous. What they are? Simultaneous processes, because they can bring in the same chamber. Though when it comes to translation here, first transcription has to happen, then only the translation can start, and the RNA has to be transported, or mRNA has to be transported outside the nucleus to that happen. That's why we use the word what? We use the word successive and simultaneous. Important words. Both of them are template depend. As a template where tRNA is used, adapter molecule is there, all those things are there. The last points are about catalyst. In this, when it comes to prokaryotes, when it comes to translation, the catalyst is 23S RNA. Here you have 28S RNA, and both of them require the same amount of energy when it comes to the process, though important portions. Again, small differences. We have talked about the differences in replication, transcription and translation, which are directly mentioned here. Let's start with the next section and go to the next part. This is a factual table. I don't have to explain this table to you. Have a small pause here. Look at the table here. This table brings out all the facts, even including the zoology sections also, into one place. So if I have to look at the facts, the numerical values wherever they are mentioned, they are all mentioned here. So here the fact is there, and on the other side, what is the explanation of that fact that is available? These facts here and there, they keep on appearing. Let me discuss a few ones, in which you have the first one, that is the genome size 5x174, you have 5386 nucleotides. Lambda fast 48502 base pairs, E. coli 4.6 10 to the power 6 base pairs. Then you have human DNA, that is 3.3 10 to the power 9 base pair. Here you have 6.6 10 to the power 9 base pair. Typical mammalian DNA is 2.2 meters in length, that is a diploid cell. Pitch of the DNA is going to be 3.4 nanometer. So in this, so they have talked about the base pairs here and all. Next one, you have base pairs per turn, that is going to be 10 base pair. Then you have the distance between the two, that is 0.34 nanometers. Typical nucleus dimension, that is going to be 10 to the power minus 6 meters. Here 10 to the power minus 9 meters is there, nanometer has to be done. And then you have a stone octamer, which have 8 molecules in it, because the octamer word is used. Nucleosome have 200 base pair in it. E. coli cell division 20 minutes. E. coli replication time 18 minutes, that is given, and the rate of polymerization is 2000 base pair per second. Poly A tail length is going to be, or polynylation is going to be 200 to 300 residues. Here in genetic code, standard amino acid 20, how many total codons? 64, how many active codons? 61, how many stop codons are going to be 3. These are the facts about the Human Genome Project. They are not exactly mentioned. This is how it is. All the facts together, don't have to worry. Most of the things are here. You can just go through them and just do the next part. Lastly, this is the table of the definitions. Now these are definitions which are throughoutly present in the entire chapter. I have taken those things and put them together in one place. Any definition you are missing, any particular word, you are doing a question, you suddenly found a word, you can come to this table and you can find that particular definition here, you can find that particular explanation here. Simple definitions are given, just use the definitions to your own benefit whenever they are available. Now, mostly we have discussed this particular part. Now the next part that we are going to discuss is going to be the NCERT itself. So I'll take you to NCERT. In that NCERT section, we are going to discuss what are the NEET questions that have appeared. I have marked important NCERT lines also. Entire chapter is important, but I have marked the lines, and we have marked the questions also, that in which year, from which section the questions have appeared. So we have hotspots of these questions also, that okay, from this particular section questions are appearing almost every time. So we'll mark those options, we'll see those things. Let me take you there, that how this is going to be. Let's start from there. In the NCERT chapter, we are starting with chapter initial section. Remember that we are not discussing the chapter, we are discussing small hints available in NCERT, where NCERT tells me, okay, I'm going to make questions from there. NEET is going to tell, I am making questions from here. Important section. Let's start with this. So this is the one. Let's start with this, the first part. The first part is where the data sets are given, match the following questions have appeared recently. It appeared in REET 2022, in which they will give these values on one side and they will ask you that which of the following is there. In the middle, what I'm going to do, I'll do small things in such a way, when I'm discussing this, if any particular question that can be formed around it, then we can discuss those also. I think it might be visible to you. I think, okay. Now, see the first question. See here. It says that 5174, 5386 nucleotides are there. Now, this is a nucleotide one and all. Now, when the word nucleotide is mentioned, can there be base pairs? So, remember that they will ask you how many base pairs are there in 5x174, 0. Hint, trick question that I can ask. Here they talk about the haploid content of human DNA, that is this one. Now, this is the haploid content. Now, if I want to give you a diploid content, which have been a question, the diploid content will be what? The diploid content will be 6.6 into 10 to the power 9 base pair, already mentioned, but this has to be done here. Let's go to the next part. Now, this is the part where many students, go fast because they think they understand the structure and all that. Very small things are mentioned. If you see this part, they have mentioned that OH of which part of the sugar is actually available in N-glycosidic bond. Now, at the same time, they use the word deoxyguanosine, cytidine, deoxycytidine. Every time a DNA part is mentioned. Now, this is the confusion that appears because of biomolecule. Biomolecules don't separate out, biomolecule chapter doesn't separate out these molecules or the cytidine and deoxycytidine. But here NCERT is very clear that when it comes to the nucleosides, the adenosine is not going to be adenosine, it's going to be deoxyadenosine. It is not going to be cytidine, it's going to be deoxycytidine, it's going to be deoxyguanosine. And remember that NCERT doesn't mention, see here it mentioned uridine, here it mentioned deoxythymidine. What it actually tells, that uridine is found only in what? Only in RNA, and deoxythymidine is present in only the DNA. So this is an important aspect. Now there is a trick question here, let's see. Two nucleotides are linked through 3' to 5' phosphodiester linkage to form the dinucleotide. Now, it's a very, very important question. Now, how it is an important question? We have done that DNA polymerase synthesize in 5' to 3' direction. Now, this is the question. Now, see the reason is given here. If you are moving from 5' to 3', the bonding or the phosphodiester bond is formed from which direction to which direction? It is going to form from 3' to 5' direction. Now, this is assertion is somewhere else, reason is somewhere else. Again, repeat, DNA polymerase is joining things from 5' to 3'. But the reason is because the phosphodiester bond is formed from which direction to which direction? 3' to 5'. So, bond direction is opposite to the direction of acting of DNA polymerase. Hidden question. In NEET, we have seen question on this. They will give an arrow here, they will give an arrow here, an arrow here, and they can give an arrow here. And they will ask you that what is this? What is this bond? They will ask, this is a phosphoester bond or diester bond. This is what? Phosphodiester bond. This is what? This is a hydroxyl bond. So, again, questions have appeared on this. Important parts. Let's go to the next section. 1869, the word nuclein. Use that table, you're done. Now, see this, in this, see how many questions. From this particular section only, adenine, thymine, because the Chargaff rule is mentioned here and all, 2022, 21, 20, 19, 17, 15. I am getting question almost every skip year, I am getting the question on that. Easy question, though a hidden question that has to be there. Remember that when it comes to the two parts, one is called complementarity of bases and another part is going to be helical structure. Now, there is a small question difference that has appeared. If somebody asks a question that Francis and Crick based their DNA double helical model on what? So, it was work of Franklin and Wilkins also. At the same time, it is the work of Irwin Chargaff also, on which it is based. But see one thing here. If they say the complementarity or the concept of complementarity It is based on whom? It was based on Irwin Chargaff. Remember that the hallmark of their paper was based on work of Irwin Chargaff. If somebody says the helical nature, it was based on the findings of what? That is Wilkins and Franklin. So remember that. Small differences there in the question, and you will have different answers which are available here. Let's go to the next part. Again, this part, this is the salient features. This is actually the characters of DNA. When you have a structure of DNA, most of you are not going to mistake in the characters of the DNA, but yes, they have made question. I can give you 5 statements and I will ask which of the following is the correct and the incorrect option. I've not seen many questions on this because in recent time no questions appeared as such, but the questions, the part is there. Numerical parts are coming from this. Now in this, so this is the previous diagram only. Only difference is going to be that they have added these bonds here, triple hydrogen bonds and the double hydrogen bonds. One question that appear, why there is an equal distance between the two? The equal distance between the two is because of what? Because the this is a purine, this is a pyrimidine. This is a purine, this is a pyrimidine. This is a pyrimidine, this is a purine. This is a pyrimidine, this is a purine. This is what makes this diagram important, that why they are equal distance, why the pitch, why the width of the DNA is not changing, because every time in front of a purine, a pyrimidine always appears. That is one. See, one question, yes, probably I have marked it here, check it here. The question is, again, let me take you to 2 questions. First question, see, what is responsible for double stranded nature? If somebody asks, double stranded nature, who is stabilizing? And I say another one, who is stabilizing double helical structure? Remember that. 2 different questions, very small. If somebody asks, who is managing double stranded nature? Double stranded nature is managed by phosphodiester bonds, because that is the standard nature, and the hydrogen bonds. They are managing the double stranded nature. But double helical nature, see the NCERT here, the plane of one base stacks over the other end. This, in addition to hydrogen bond, confers stability to helical structure. It doesn't say double stranded structure. So helical nature is because of what? Helical nature is because of hydrogen bonds and the stacking force. So double stranded nature is because of phosphodiester bonds and hydrogen bonds, but double helical nature is because of hydrogen bonds and stacking force. These are the important questions. Let's go to the next part. Here, Central Dogma have made questions so many times. 2021, 13, at that time NEET Karnataka was separate to 2010. Good number of questions have appeared. Question will be appeared. They have given one question, that which of the following doesn't follow the central dogmas, and the answer was HIV, because HIV shows reverse transcription also. So that was the question. Important questions are appearing from this particular section. Let's go to the next part. Okay, packaging of DNA, important question 2022. Again, we have discussed this, as how many meters are there. Some numericals are also in line for this, and then the nucleosome section, chromatin. There's one thing here in nucleosome, we have chromatin word given, in which beads on string word is used and all that. Problem is that according to NCERT, the DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. The typical nucleosome contain this. Nucleosome constitute of the repeating units of structure of called chromatin. The problem is, in cell chapter there is a small issue. In cell chapter, chromatin is shown to have RNA as well. So that is also mentioned there. Remember that. In here, nucleosome is seen as a unit of chromatin, and it is having only DNA. This is negatively charged and positively charged. Histone octamer is there, though when it comes to the cell chapter, you have RNA and other things, or non-histone proteins are also involved in that. H1 histone is not a part of the octamer. Remember that octamer have H2A, H2B, H3 and H4, but H1 is not part. H1 is seen in the chromatin, but is it part of the is the part of the octamer? No, it is not part of the octamer. Important point, don't forget that point. Again, I am just going to on the basis of the questions. Important points are there about nucleoid, loop structure, arginine, histidine, all those things are mentioned here. Okay, there you have 2025. The recent question appeared from this particular section. So this was the question about the chromatin nature. A question appeared that so this was a question recently, important part, heterochromatin, euchromatin. Don't forget to look into it. Again, that table is there. It will give you all the definitions. Now the next part is search for genetic material. Recently a question appeared from this also. 2024 also you had a question, important part, Frederick Griffith. That means I told you, out of all the experiments are covering Griffith experiment, or a matching actually came in which you have to Griffith and other scientists were given. You have to join the scientists. Important portion. What are the bacteria involved? Who's the R strain? What is the S strain? All those parts have to be done. Again, it's important schematic. If I want to give a question to you, I will not give you this, I will not give you this, and I will ask you that what is the result, what is the expected result there. Those kind of questions can appear in this. Let's go to the next section. Again, experiment is there, important section. Now let's go to the biochemical characterization part. This is this here, remember that? They will ask you that which of the chemicals are purified and what are the enzymes that have been used. So RNases and all that is mentioned in Avery, McLeod, McCarty. Now here's a confusing line there that the DNA caused the transformation. This concludes that DNA is a hereditary material, but not all biologists were consumed. They did prove that DNA is a genetic material or the hereditary material, but was it the unequivocal proof? No, it was not an unequivocal proof. Now the next one, the most asked question from this particular section, that is DNA genetic material. That is actually Alfred Hershey and Martha Chase experiment. So many times the matching is coming from them. Important sections. Now, if you see, the diagram is actually this one. So many students remember this diagram, this next diagram that is given, infection, blending and centrifugation. Though many students don't read this section, that's why I specifically marked, and questions have been coming on that. In 2023 Manipur and the REET 2022, I have seen this question. This is actually the first step. What students do, they remember the first step is going to what? First step of this. The next one, in which they talk about the bacteriophages. That this bacteriophage is there, this bacteriophage is there. But in reality, how these bacteriophages are made? How did I make a S35 labeled bacteriophage or a P32 labeled bacteriophage? That is here. That is given in NCERT. It is mentioned here that first you make the E. coli radioactive, then you grow a normal virus over it, then virus eat the bacteria and they become radioactive and so on. So that part is missing in many of these books, or many of these students don't read this. Remember that this is the first step. First is the production of the viruses, and then you have the next part of the experiment, that is the second phase of the experiment. Sometimes remember this experiment have another name because viruses are used and you are infecting viruses with this. So this can also be named as transduction experiment. Important experiment, infection, blending, centrifugation. Now, what is the key event that actually proves that DNA is a genetic material? That is transfer of radioactivity. What is the word? The word is transfer of radioactivity, which proved that DNA is the genetic material because transfer of DNA happened. Let's move to the next section. Then you have properties of genetic material. Now NCERT suddenly jumps into the next section where they talk about the properties of genetic material. This have appeared before, that which of the following have RNA genetic material, that is the tobacco mosaic viruses, QB bacteriophages, other viruses can also be there. Here in 2016 a question came on this. What are the expectation from a genetic material? Now what is the first expectation? To generate its replica. Now this is a hidden question here. It should be able to generate its replica. Can DNA do it? Yes. Can RNA do it? Yes. Both the answer is yes. Important part. Chemically and structurally stable. DNA is. But is RNA stable? No. RNA is not stable. Provide scope for slow changes. DNA and RNA both allow scope for changes, but the question is slow changes. RNA is fast changes. And then you have express itself in the form of Mendelian characters. Both of them have that. Remember that DNA can express itself in the form of proteins, but with the help of what? With the help of RNA. Remember that. Can DNA directly express itself? No. DNA can indirectly express itself in the form of RNA. That's important. So who does direct expression? RNA. Who does indirect expression? That is DNA. Go to the next section. Okay, this I've deliberately marked. This is the part that explains the DNA versus RNA, which is how DNA have more stability, that is about the 2-OH position, that is in ribose sugar but not in deoxyribose sugar. It talks about the position of thymine, which is more electropositive, uracil is not electropositive. It does talk about that RNA has more mutative nature, they have a catalytic nature, that's why it is more reactive. All those portions are here. Don't forget to have a look at it. Very, very important sections. Okay. Before the RNA world is a small topic, but it has made questions. Now, which of the following parts show evidences? Metabolism, translation, splicing are evidences of RNA world. Remember that one hidden evidence which will come later on, that is in translation section. That evidence is the translation, in which you are talking about what? You are talking about 23S rRNA, and here splicing you have snRNA that is actually doing that, which is available here. Next portion. 2015, 23, 22, 15 questions have appeared on these particular lines, that how DNA has evolved from the RNA, how RNA is a central molecule, is still a central molecule in most of the metabolic processes. Next part is replication part. Here in this section, they first talk about the semi-conservative nature. This diagram, I can give you this diagram and ask you that what does the diagram show? It shows semi-conservative nature. Remember that Watson and Crick model for semi-conservative nature. So the NCERT is telling you here that semi-conservative nature is actually proposed by whom? It is proposed by Watson and Crick. Next one is the experimental proof, how of the semi-conservative nature, Meares-Stahl experiment. I have planned some numericals for you that there we will discuss them, that what is going to be. Now, one thing, a small thing that I wanted to point out. When students see this, the thing is that they see this tube, this is a cesium chloride gradient that they are seeing, and they are giving gravitation force there. Remember that. If I want to give a question, I can make this tube like this also. Means I can make a straight tube also. So if you see this tube, so remember that the lower portion have more gravity, upper portion have less gravity. So when those kind of questions are given and all that, remember that I can straighten the tube also and give you that particular part. Here one thing, here a hybrid DNA is given. Here let's say you have 100 DNA molecule, here it became 200. But see, all 200 of them are what? Are the hybrid DNA. So if I want to give a confusing question, I can give 100 molecules here and I will not make this. I will make only this, because all of them will convert into this only. But I can write the different numbers. So sometime this part is given, sometime this part is not given. So that is one time, one question appeared like that in our old work, that you should not be worried about. Though we will discuss a question type on this, what kind of questions I can expect from a Meares-Stahl experiment. 100% question appearing every year. 2016, I have the question on VCF-ABA, Taylor et al., what they are doing, though Taylor et al. Has appeared in matching in other questions as well. In 2024, I have enzymes which are involved in the replication. Here we are talking about this. Here DNA polymerase are the important one. DNA are template dependent, which are doing that. Remember that most of the things are template independent in this chapter. Again, small points from the discontinuous part. We have already discussed that section. From origin of replication 2024, means replication is a question that is appearing in recent exams. A very important part, that is the replication fork. Now one hidden question here. When we are discussing this, so this is the template. So this is the, if you see this. Now many students see that how can I get that this is the template and all that. Remember that you are opening the DNA from this side. So if you are opening the DNA from this side, many students start to read from here. No, you cannot read from here. If you are opening the DNA from down to up, then you have to read the DNA from down to up also. So this side, which is the continuous synthesis is happening, here the template type is 3' to 5'. Here template strand is 5' to 3'. So on 5' to 3', you have continuous synthesis. On 5' to 3' template, you have discontinuous synthesis. Very important section. Now, when they see this, so when you are opening the DNA, here the synthesis is happening. If you see the synthesis of DNA or synthesis of addition of nucleotide is happening towards the opening of replication fork. Here it is happening away from the rest. So in case of discontinuous strand, we'll see the synthesis is happening away from the replication fork. This is towards the replication fork. This is a small hidden question that is available here. Now transcription, easy portion. This is a very important paragraph, this one. Why DNA, both the strands of DNA are not coded? NCERT have given two reasons. First reason was given as that it will further complicate the process, and the other reason is given as that it will make the same, or they will make a RNA which will actually connect to each other, and they make a double strand RNA and it will become useless. That is what is given here. Don't forget this paragraph. We are going to transcription unit, what is available, promoter, structural, gene and terminator, and with this you have this. Now see the importance. From this coding, non-coding, this part, I have question 2024, 23, 22, 19, 18, 14, 12. I have so many questions appearing from this. There are certain conceptuals coming from this section. I marked those conceptual. Towards the end, we'll discuss those conceptuals also. We have made a separate video for them also. You can go into that and you can see that how they are. Let's start with the next one, or let's go to the next one. Again, this diagram have appeared. Please make right now in front of me, make this that you are burning this image into your head. When this is given, do it like this only, that whenever the diagram is given, I am looking at this diagram, and every time I am looking at this diagram correctly only. Very, very important section. Though this itself, the entire thing is structural gene, but NCERT made written structural gene here, template here, and the coding strand on this side. Just make this mental note. Very, very important section. Let's go to the next part. Again, 23-24, transcription is making questions regularly. Let's go to this important section. Polycystonic and monocystonic genes, what are exons, what are introns, what are their implications, this one. Now I personally feel introns, exons are there in the other portion. You can actually remember them easily. Monocystonic, polycystonic, I'm not going into the semantics of them. Just see this. This paragraph has not made questions in NEET yet. In old papers it have appeared. So inheritance of character was also affected by promoter and regulatory sequence of structural gene. Hence sometimes regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA and protein. So this gives you the idea why the cistron term was needed. Somebody can ask a session that gene and cistrons, or why the cistron term is needed when gene term was already there. Gene term has lost its value because any part of DNA which is doing anything, that was named as gene. That's why we have to come up with the concept of cistron. Though important paragraph, don't forget to remember this paragraph. Remember it line by line. In 2016, I have question on DNA dependent RNA polymerase. They asked that what is that polymerase and where it is working. Now see this. Now here you have the sigma factor point mentioned, here the elongation point. See, so many students think that sigma factor is involved here. Sigma factor is not there. Sigma factor is actually left. They are showing the lefting part in this, and then you have the rho factor that is available towards the end. Remember that promoter and terminator are required for this. Now come to the most important portion, that is sigma factor and rho factor have been making question in recent time. 25, 24, 24, 22, here 2021 and 2020. They have made questions. Important part, remember that intriguing question is that how a polymerase catalyze all the three steps: initiation, elongation and termination at the same time. So and here it feels like the initiation, elongation, termination all are made by RNA polymerase, and as we go further, here it shows RNA polymerase to initiate or either terminate, it need what? Sigma and rho factor. So remember that it can actually catalyze all the steps, but for initiation and termination, it needs help. But for elongation, it doesn't need any help. Opening of the DNA is also explained. Remember that all the processes are done by whom? RNA polymerase itself. Nothing else is required for this particular process. Questions have appeared that in transcription, who is causing unwinding of DNA? Now the unwinding will come, students directly jump to unwind days. No, You don't have to jump to amine-base. If it is transcription, it is always going to be what? RNA polymerase. Now NCERT doesn't explain that what is happening here, though it does talk about post-transcriptional modification, in which it talks about capping, it talks about pollination, that is also called tailing, and it talks about the splicing portion that is happening here. They are given. Let me show you the importance of these sections. How important are these sections? It is clear in the next slide. See the questions. RNA polymerase 1, 2, 3, what they are making, it's a continuous question. 25, 24, re-24, 23, 21, 19, 16, 12. Super hot spot, a very much focused question. I will expect a question this year also from this particular section. Second part about splicing, capping and tailing, 2022, 17, 12 and 10. Remember that I have taken questions only up to 2010. More questions have appeared before exams also. Very, very important section. Don't even think that you can do things without it. Though I just want to make something because which have not appeared, but it will come later on. The presence of introns is reminiscent of antiquity. Line from NCERT, good line. It has a potential of a question, and at the same time it also talks about the dominance of RNA world. Never forget these two lines also. Mark these lines in your NCERT and we'll go forward. Next one is genetic code. In genetic code section, I'll take you small portion. What kind of scientists actually worked? So physicists, organic chemists, biochemists and geneticists. These are the scientists which were there. Who proposed a triplet? That was George Gamow. George Gamow is a recent question, 2024. The question, a small kind of numerical sort of thing came from George Gamow part. We'll discuss George Gamow as a separate question. Very important concept is there. Then the discovery of genetic genetic code, that is Hargobind Khurana. Then you have Nirenberg and you have Severo Ochoa. All these persons, we have already discussed them in the table before. Important people are there. They have given good number of data sets. So just have a that who did what, we have already discussed that. Genetic code I am going to discuss in a separate video. The genetic code has made some things. I'll discuss genetic code in a separate thing. Though if you see genetic code table 2023, 2019, 2013, 2010, we have got questions from this particular section on a regular basis. Next one are mutations part. Mutations, frame shift mutation are mentioned. Here sickle cell anemia is mentioned, which is a kind of transversion. It is a kind of point mutation. Though the important part of the questions that have been made, they are made by this section about the frame shift mutation. In the frame shift mutation or deletion mutation, what is happening, how the frames are affecting things, there are certain numericals around it. We will discuss those numericals towards the end also, but never forget to see this line, mark this line, important parts. A raptor molecule, now this is what I was saying, that if you see the very old book, you will see a difference in the diagrams. Here you have inverted L model given by Kim and Clark, but in the old models you might find a model that is given something like this. But that is not there anymore. It is given Kim and Clark model, important parts. The portions are given about this. tRNA molecule, what is the acceptor and what is the anticodon loop and all, they are all mentioned here. Easy sections, but again, have a read about them. Next one is about translation itself, how translation is working. Now see, I want to show you a specific question. Now see this. Here, this is the first phase. The first phase is what? Activation or amino acylation of tRNA or charging of tRNA. That is first phase. But did the translation initiated? No, it is not initiated. As we go to the next chapter, next lines, we'll see something like, just see this. See this. In this section, when the translation is explained, see this, when the small subunit encounter an RNA process of translation of the protein begins. Now, beginning is here. This is 2025 question. And just see the previous one. Both of them appeared in NEET. This is the 2020 question. Small word change and you have a different question. First phase is mentioned, go for charging of tRNA or amino acylation. But if they say beginning, starting, then you have to go for the mRNA binding to the small subunit. That is an important aspect there. UTRs have also made a question in recent times, in which why they are required. They are used for efficient translation. They are available where release factor I've already discussed in the previous section. Then you have regulation. 4 levels are given. Remember that these 4 levels are eukaryotic levels. Prokaryotic level, only transcription level is mentioned in NCERT, no other level is mentioned. And at the transcriptional level, you have mentioned the Operon systems and also here Operon system is explained, definition of Operon in 2018. In 2024 elucidation part was mentioned, a small matching came on the Jacob and Monod. See this part, Lac Operon, what does Z do? A or ZYA, what are these things doing? 24, 23, 22, 22. 22. Why I am saying? Because 2 times question come in. There is a separate 2022 exam, 19, 13 and 10. Very, very important question. Very, very centric question. It is going to appear. I do believe in 2025 it has a high chance, 2026 that it has a high chance to come. We'll discuss them, though these diagrams have also appeared, that how the repressor works, how this works. They have also made question 2023 Manipur, 2013 and 2015 have also been asked. Now what is inducer, who is an actual inducer, those part have also come in 2023, 2016 and 2013. All the hotspots are marked. You have to just go through this video, you will understand all the parts. We'll discuss the negative and positive regulation in a separate video. Next part is Human Genome Project. Human Genome Project is part of zoology. We are not discussing that. I am discussing till here only. Now just to show, we have done this much, we have done most of the parts. So in this video, we are starting there. There are other shorter videos available in this particular series in which you can go and you can look for a specific topic of genetic code and other portions. And numericals were also discussed in this. Don't miss them out. It's an important aspect that is mentioned. So this is all in this video. I hope that molecular biology is done for you right now. Let's see how much it is. Okay.